Okay, so today we are going to work with our codons and going from DNA, whether it is the coding strand or the template strand, and making that into an amino acid or a polypeptide chain. So, we went over last time that our codons are going to consist of three nucleotides. So, just like in DNA replication, when we were doing that last chapter, if you remember, DNA polymerase number 3 moved in a 5 to 3' direction on our new DNA strand. We are going to be reading our mRNA in that same direction to make our amino acid chain. So, if you think about mRNA, we have 4 different bases we can use. We have adenine, cytosine, guanine, and no longer thymine, but uracil. So we have 4 different ones. Now, if we have a codon, we talked about it's going to be 3 nucleotides. So if we have 4 of our nucleotide choices, what we're going to do is, since we're making 3, we are going to cube that, or we need 3 nucleotides. So what you do here is take how many bases you have, which is 4. We have 4 possible choices, and we're going to put 3 of them together. So it's simply 4 times 4 times 4. 4 times 4 is 16 times another 4, which ends up being 64. So we have 64 choices of codons. In other words, we have 64 different possibilities we can have when we put these 3 nucleotides together, given that we have 4 options, all right? If we had 5 nucleotides, it would be 5 times 5 times 5. So if we had 6, it'd be 6 times 6 times 6. If we needed a codon of 4, which it never happens, but it'd be 4 times 4 times 4, and just add another 4 at the end, okay? But anyway, we have 64 codons. Each of those codons is going to translate to an amino acid. Now, some codons, sorry guys, some of our codons might go to the same amino acid. It's going to happen frequently, just because we only have, if you remember, 20 amino acids. But anyway, some of our codons are going to go to the same amino acid, but we are never going to have a codon go to more than 1 amino acid. All right, so it'll always only go to 1. So here is our chart, and this is going to be mRNA we're using this chart for. And this is going to give us our amino acid. Okay, so let's say our DNA for our template strand, and I'm just going to use a very small one here. Let's say our DNA is A, G, C. No. Let me make that 3' there. Say this is 3', this is 5'. Okay? Our mRNA, this side would be 5'. It always says, the opposite. A would go to U, G would go to C, C would go to G, and then this would be 3'. So we're going to read this 5' to 3', always, always, always. And I'll give you guys some variations in this just so we can get comfortable with always going 5' to 3'. But, so here's how we use this chart. UGC. Our first letter is U. So on the left side of our chart here, we're going to go to the U. The second base is C. C is going to be up top. So we know U, it's going to be in one of these 4 boxes. The second one is C, so we know it's going to be in one of, or only that box there. Now our last letter is going to be G. That's on this side, so that tells us, you see G right here. And our abbreviation for the amino acid is SER. And SER, the full amino acid name is serine. You do not have to know the full amino acid names. Don't worry about memorizing them. They will give you either the on the AP test, they will either give you the full name or the abbreviation of it, but you won't be needing to know all 20 of them specifically. All right, let's do something a little bit harder here. Let's say our coding strand, our coding DNA is, let's say, T, T, G. And let's say this here is 3' and 5'. Okay, so where things is going to get a little tricky. So our coding strand, we first have to go to our template strand of DNA. So T goes to A, T goes to A, G goes to C, 3', it would be 5' on this side, 5' would be 3' on this side. So now let's go to our mRNA, because we remember guys, the template strand is always going to mRNA. We're going to start off, this is going to be 3' now, it's going to be opposite of that. T or A goes to T, but it's mRNA, so it's going to go to U, U, and a G. This is 5'. Now here's where things are very easy to mix up. So you always want to start with the 5' side when you are translating. So it's actually, even though it says 3' UUG, we would actually code this, and I'm going to flip it just to make it easier for us. It's 5' GUU. So we want to start on this side with the G. U is the second one, and U is the third. So if we come over here, GUU, it would be VAL or valine. Okay? So this is one amino acid chart, guys. I like the second one better, which is this one. This one, you are starting in the middle. So if we want to do the same one, the GUU, oops, sorry. If we want to start with the GUU again, starting in the middle, G, go to the next circle, U, and the last one, U, and it turns out to be the same amino acid, valine, okay? I like this one better, but usually on the AP test, they usually, unfortunately, give you this one, all right? Okay, so that's it with coding, guys. We're going to go over how transcription and translation happens, but that stuff there is just how you code getting from DNA to mRNA to an amino acid. The two main things to look out for when coding is, are they giving you the template strand or are they giving you the coding strand? You always need to transcribe with the template strand. If they give you the coding strand, go to the template strand first. And the second thing is to watch your 5' and your 3'. Remember, when we are putting things into that amino acid chart, we always have to start with the 5' end of mRNA and not the 3' end. Okay? So, going on to the next section, guys, the genetic code, it's pretty universal. Every single organism is going to share the same bases. It's all going to be for DNA A, T, G, and C, okay? So what this means is, since they are pretty much the same, we can actually use a lot of the same genes and take them out of one organism and put them in another, and yeah, they'll be usable. That video didn't work too well, guys. It's bioluminescence. I don't know if you guys have ever seen in Disney World, they have Animal Kingdom, they have Pandora, and at night you can go there and it's like bioluminescence everywhere. Now, in reality, it's just, glow-in-the-dark paint. They really don't have bioluminescence on it. But what bioluminescence is some organisms like jellyfish or lightning bugs, fireflies, or if you guys have ever seen the anglerfish from Finding Nemo with the little thing on the front. But anyway, so all these organisms have bioluminescence in them. And what we can do is we can extract that gene for the bioluminescence and put them in something else, put them in another organism, okay? So this one here is a tobacco plant, and they put the firefly gene into it, so that makes it glow at night. Here's a pig, and they put the jellyfish bioluminescence gene in it, and you can see the nose and the feet have a glow to them. So that's how we can use these genes, because they are universal, and almost transplant them from one organism into another, and they'll be fine. Okay, so we already went over transcription. We went over the small process of it, and we went over coding, going from DNA to mRNA. Now we're going to talk about how this actually happens. So just like our DNA, when we're going from DNA to DNA and DNA replication, we're pretty much going to do the same thing with RNA. So if you remember, we started off with our RNA primer, and then after that, we attached our DNA polymerase to it, and the DNA polymerase walked down the DNA and added new nucleotides to it, and it went 5' to 3'. For making our RNA, it's the same exact process with a very few differences. So if you remember, we needed that RNA primer at the beginning for DNA polymerase to latch onto. Now for this one, we're starting with RNA, so we don't really need a primer. RNA polymerase can just latch on and start doing its thing, and it's still going to move 5' to 3'. And I'll post that video for you guys on transcription. So here is the bubble, I guess you could say. And you guys will see this really well in the video. But there's the little bubble being made. And then the RNA polymerase will come in, and it's going to start making the DNA 5' to 3'. So the 5' end just keeps coming out, and the 3' end is going to be at the replication fork there. When it is done being made, or RNA, it's going to just be done. It's going to close that bubble, and then our RNA polymerase is going to leave. And that's pretty much it. So we now have our RNA. Okay. Also back here, guys, there is a promoter region. I forgot to mention that. So the DNA sequence where RNA polymerase attaches, that's called the promoter. In bacteria, so at the end of the sequence, so at the very end, there's going to be a terminator, and I don't mean the movie. And all that's going to do, guys, is just terminate our whole transcription process. It might be a little bit before the end of the DNA. Why? Because we don't need all the DNA. We don't need all of our DNA in order to make the protein. Some of our DNA, especially at the ends, we talked about the telomeres last chapter. Okay, that's going to be a non-coding region. It's not going to be used to make a protein, so we don't need it. So the terminator will be before that area. So there's 3 stages of transcription. And we'll go through these pretty quickly. There is initiation, elongation, and then termination. So we're going to have a start point. These are where our promoter is going to be. And it's going to be not all the way at the end of the DNA, but it's not going to start exactly where it needs to. We're still going to have some kind of cap at the end of it that's going to be made out of some nucleotides, okay? So the RNA will be, I don't want you guys to think the whole thing is going to be coded for our amino acids and our proteins. We are going to have some nucleotides, just like its fillers in at the end. All right. We're also going to have some transcription factors, and this is going to be where our RNA polymerase can bind, and this is where our transcription is going to begin. So just a funny story. When I was in college, guys, I'll never forget the day I learned about the transcription initiation complex. And when we were learning about it, they were talking about it's a TATA box, and it's TATA. So, sorry for the bell, guys. So the TATA box is going to be the beginning of the initiation complex for transcription in eukaryotes. And all it is it's obviously it's TATA, so it's not going to be RNA, it's going to be DNA. It's on the DNA sequence, and that's going to be where the RNA is going to start transcription. So I'm like, oh, okay, so TATA, and I was all good. And I understood where it started at. So I'm at Duquesne University. I walk outside, and you'll see when you guys get to college, there's like tons of different things going on in the main area. And I see this sign that says, and I was pretty naive at the time, but it said Save the Tatas. And they were selling t-shirts about like Save the Tatas. And I'm like, how did they know? I just learned about the Tata box in transcription, in class in biology. And I was just like, wow, they had this college is pretty good. They got it going on, Found out later that it was a completely different meaning for Tata. So it was, talking about breast cancer and stuff like that. But it was just kind of funny how things worked out. But anyway, so the Tata box, this is where the initiation complex is going to be for eukaryotes to start our process of transcription and making our mRNA. Okay, so we did that picture. So here is our Tata box, okay? And that's where our mRNA going to start to be made. Now, RNA polymerase is going to move on the RNA 5' to 3'. If we're talking about the DNA, it's going to be moving the opposite way, which would be 3' to 5' along that strand. But anyway, so for the RNA polymerase, we're going to be adding nucleotides to our RNA at about 40 nucleotides per second. Okay. Now, as the RNA is moving, just to kind of go back for our DNA replication, we are still going to need to break apart our DNA. So we're still going to need helicase in there to break those hydrogen bonds between our nucleotides in order to get the mRNA made. Okay, so and that's pretty much the only character we still use from DNA replication is our helicase. And again, that's just to break these two strands apart here so that the mRNA can be made. And you guys can see it's real quick. It goes like this. They attach that strand, and then it comes out on the left side here, and it detaches. And after it detaches the mRNA, then our DNA can rebond together. All right. So for bacteria, our polymerase stops transcription at the end of the terminator, and everything is done. Nothing else needs to be modified for prokaryotic cells. For us, for eukaryotes, there's going to be RNA polymerase number 2, and at the end, we're going to have something called a polyadenylation signal sequence. This is also called the poly-A tail. And what this is, guys, very simple. It's about 10 to 35 nucleotides past our end of like whatever we're using to make the amino acid. But all it is going to be a bunch of A's, okay, a bunch of adenines, A, so on and so forth. And that's going to be the end of our mRNA. So that's going to be on the 3' side of our mRNA. We're just going to have a bunch of A's at the very end. And that's going to be a non-coding part of RNA or mRNA. So it's not going to be used to make an amino acid. All right, we're going to do a little bit of modification for eukaryotes, and then we'll call it a day. So after our RNA is transcribed, we are going to add a few things to it. We already talked about the poly-A tail on the 3' end. We are also going to add a GCap on the one side. It's going to be guanosine. It's called guanosine triphosphate, but it's just going to be a bunch of guanines. So we're going to have a cap, a GCap on the one side, just a bunch of G's, and on the other side we're going to have a bunch of A's. So if one of these is missing, this is important for you guys to know, if we don't have the ends, if we don't have, whether it's the GCap or the poly-A tail, our mRNA is not going to be used to be making a protein, to be making an amino acid sequence. Instead, if you remember last chapter, we talked about the nucleases and exonuclease. We are going to once again use these in order to digest our mRNA. In other words, break it apart so we could still use those nucleotides to later make more mRNA. All right. And I'll post that video for you guys. So there is your GCap on the one side. There is your poly-A tail on the other. Okay. So we're also going to have different parts of our mRNA. Now, we are not using all of our DNA to make this mRNA. We're using a part of it. All right. We're going to be using a certain area that we need to make a specific protein. So we need to get a protein out of our DNA, and we're just going to target the area of that DNA that makes the protein. However, all of that area of DNA, that specific area, might not be used to make the protein. So let me try to give you guys a visual on this. So, and I'm going to shorten this a lot, so don't think it's this short. But let's say this is our DNA. Let's say this area to this area right here is what we're going to need to make our protein. So what we're going to do is we're going to take that area out. And that is going to be our mRNA. All right. Now, in our DNA, if I go back up here, let's say that only this area right here, this area right here, this here, and this here. Let's say those are going to be the areas that we need to make our protein. So if we come down to the mRNA, it's here, 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 and here. These little red areas I have left over, they still got transcribed. They're still going to be in our mRNA. But this area here, this area here, this area here, and this area at the end, we don't need them to make our protein. So we do not want to use the energy to make them into amino acids, okay? So there's a little picture. Hopefully you guys can better see what I'm explaining here. So the areas, the black areas there, that are going to be translated into amino acids, we call those areas our exons. All right. And then our other areas, the areas that are not going to be used to go into amino acids because we don't need them to make the proteins, those are going to be called our introns. Okay. So what we're going to do is we're going to use something called RNA splicing because we don't need those introns in there. Why translate them into amino acids if they're not even going to be used? So again, guys, we're just going to remove those introns. Okay. So here is our whole part. You guys can see the introns here. We call this PreMrna, and I know you guys saw it in the pictures before, but I didn't mention it too much just because I knew we were going to get to it later on. So this is our PreMrna. It still has the 5' cap on it, the GCap, and it has the poly-A tail. It has the exons in our darker red and our introns in this peach color, I guess we could say. When we are making our actual coding mRNA, we are getting rid of those introns because we don't need them. Okay, guys, I think that's enough for today. We'll finish the RNA part tomorrow. Have a good day.